Assalamualaikum warahmatullahi wabarakatuh. My name is NAJAH, and today our group want to present about homeostasis. This is content of this topic. What is homeostasis? Homeostasis is the ability to maintain a relatively stable internal state that persists despite change in the world outside. All living organisms must regulate their internal environment to process energy and ultimately survive. First, homeostasis results in a dynamic equilibrium where continued change keep on taking place and yet steady conditions are maintained. Second, homeostasis is mainly involved in managing various internal variables of the living system like body temperature, pH of various fluids, the concentration of different ions, and the body sugar level. There are three types of homeostasis that we can learn. First, thermoregulation. Thermoregulation is the process occurring inside the body that is responsible for maintaining the core temperature of the body. Thermoregulation works by the negative feedback loop, where once the body temperature is either increased or decreased beyond its normal temperature, it is brought back to normal. Second, osmoregulation. Osmoregulation is the process of maintaining a constant osmotic pressure inside the body by balancing the concentration of fluids and salts. During this process, excess water or ion or other molecules like urea are removed from the body to maintain the osmotic balance. And lastly, chemical regulation. Chemical regulation is the process of balancing the concentration of chemicals like glucose and carbon dioxide in the body by producing hormones. During this process, the concentration of hormones like insulin increase when the blood sugar level increase in order to bring the level back to normal.
Assalamualaikum warahmatullahi wabarakatuh. My name is BATRISYIA binti HALIZAN, and I'm going to explain on how the homeostasis is working. This mechanism will tell us on how the body internal changes the entire environment. It requires three interacting components. The first one is receptor. It is a nerve ending that reacts to a change such as heat or cold in the body. The second one is control center. It is a body structure that determines the normal range of the variable. The third one is effector. It is the cell, tissue or organ that responds to signals from the control center. Thus, it provides a response to the stimulus. From this figure, we can simply explain on how the homeostasis is working. When our body is at normal state, there are in balanced state in homeostasis. After that, when there are changes of the temperature of our body, it will be detected by the receptor or the sensor. This information will be sent to the control center, which is basically the brain. Then, the control center will send the information to the effector to respond to the changes. This response will give feedback to influence magnitude of the stimulus to return back to homeostasis. Feedback mechanisms. Feedback loops are biological mechanisms whereby homeostasis is maintained. This occurs when the product or output of an event or reaction changes the organism's response to that reaction. There are two types of feedback mechanisms. The first one is negative feedbacks. This feedback is to reduce an excessive response and keep a variable within the normal range. For example, body temperature regulation and control of blood glucose. The second one is positive feedbacks. This feedbacks is to intensify a response until an endpoint is reached. For example, blood clotting and childbirth. The negative feedback loop is the most common regulatory tool that is being used to maintain the homeostasis. There are situations where the negative feedback loops will occur. The first one is human body temperature. The hypothalamus of a human reacts to temperature fluctuations and responds accordingly. If the temperature drops, the body shivers to bring up the temperature, and if it is too warm, the body will sweat to cool down due to evaporation. The second one is human blood pressure. When blood pressure increases, signals are sent to the brain from the blood vessels. Signals are sent to the heart from the brain, and heart rate slows down, thus helping blood pressure to return back to normal.
Assalamualaikum warahmatullahi wabarakatuh. My name is HUDA binti NORFHARIZAL, and today I will continue to present about osmotic concentration in blood. Definition. Do you know what is the osmosis? Osmosis is the diffusion of water molecule across a semi-permeable membrane from an area of lower concentration to an area of higher concentration solution. Next, osmotic concentration, formerly as known as molarity, is the measure of solute concentration. Osmoregulation is the process of maintaining salt and water balance across the membranes within the body's fluid, which are composed of water plus electrolyte and non-electrolyte. How osmotic concentration work in blood? Okay, when a red blood cells are in the hypertonic solution, which is a higher concentration, the water flows out from the cell faster than it comes in. Next, a red blood cells that is hypotonic solution, which is lower concentration outside the cells, will result in more water flowing into the cell than out. Okay, the last one is when a red blood cell in isotonic solution, the flow of water in and out of the cell is happening at the same rate. Okay, now let's relook at the picture in this slide. Okay, the first picture is when a red blood cell in hypertonic solution, so the cell will be clinician of the blood cells. And the next picture is when a red blood cell in isotonic solution, so the flow of water in and out of the cell is occurring at the equal rates, which is it will be equilibrium. And the last picture is when a red blood cell in hypotonic solution, so swelling of the cell and hemolysis of the cell will happen. Hypertonic solution is a greater concentration of solutes than another solution. When cell is immersed in hypertonic solution, osmotic pressure tends to force water to flow out of the cells. In order to balance the concentration of the solutes, either side of the cell membranes. Next, hypotonic solution is the lower concentration of solutes than another solutions. For cell without a cell wall, such as animal cells, if the gradient is large enough, the uptake of excess water can produce enough pressure to induce cytolysis and rupturing of the cell. Okay, the last slide from me is isotonic solution. A solution is isotonic when its effective osmol concentration is the same as that of another solution. Water molecule freely through the plasma membrane in both directions, as the rate of water diffusion is the same in each direction, so the cell will neither gain or lose water. So that's all from me, so I will pass the slide to Aizuddin.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Aizuddin bin Zaidi, and I would like to continue on the presentation on how homeostasis is maintained inside a freshwater fish. So first of all, we need to know that there are two types of fish, which is the first one is saltwater fish and the second one is freshwater fish. Saltwater fish lives in an environment which is high in salinity of water, where meanwhile freshwater fish lives in an environment which is low in salinity of water. Both saltwater fish and freshwater fish are mostly stenohalide animals because they cannot tolerate substantial changes in their external osmolarity, or in other words, they cannot tolerate a wide fluctuation in the salinity of water. If the two are to switch places, neither both of them can survive any longer and will die due to they cannot tolerate in different environments. Next, we will focus on the osmoregulation process in a freshwater fish. So first of all, freshwater animals are hyperosmotic to freshwater. What is meant by hyperosmotic to freshwater? So the meaning is the concentration of salt in freshwater animals is higher than their surrounding environment. So what did they do? They constantly take in water from their hypoosmotic environment. Freshwater fish also lose salts by diffusion process through their skin. To maintain the water balance inside their body, freshwater animals do not drink too much water compared to saltwater animals, but instead they produce or they excrete large volumes of very dilute urine from their kidneys. Since freshwater animals lose salts from their body by diffusion, the salts that are lost needs to be replaced, and it will be replaced by the foods that they eat, and the second one by salts uptake across the gills by active transport. Here are some examples of freshwater fishes. As you can see in the slides, there is brown trout, bluegill, catfish, largemouth bass, and anglefish. So that's all from me.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi ta'ala wabarakatuh. I am NURUL HAYATI binti ISMAIL. Want to present to all of you about the importance of osmotic concentration. Firstly, the osmoregulation requires energy. The osmosis tends to equalize the ion concentration. For the example, since the fish required different ion level from the environmental concentration, they need more energy to maintain a solid gradient that optimizes their osmotic balance. The energy required for osmotic balance depends on the multiple factors such as including the differences between the internal and external ion concentration. And next, when the osmolarity difference are minimal, the less energy is required. Freshwater animals maintain concentration of salt. In order to keep the internal environment stable, continuous adaptation with regard to temperature, pH, concentration of sodium, potassium, calcium ion, carbon dioxide, and oxygen take place. Water enters their body due to osmosis because the salt's concentration inside the body is higher in surrounding water. Without any active regulation of this process, fish would swell and get more bigger. Their body produces a large amount of urine and it lost salt at the same time. The conclusion: the homeostasis is the tendency towards the relatively stable equilibrium between the interdependent elements. The solid concentration and size of molecule affected the movement through the semi-permeable concentration. The water moved from the high concentration to the low concentration. It means the high concentration is hypertonic and the low concentration is hypotonic. And the kidney is reabsorbed salt before it ejected to decrease the loss and activity of salt by using the gills from the urine. And last but not least, the freshwater fish produce more volume of dilute urines that is low in the salt. That's all from me.
These are the references that we use to search for the information regarding the topic. So that's all from our group. So thank you very much. Assalamualaikum warahmatullahi wabarakatuh.